Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing next question paper for the biology section. This is session 2. This was conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of next exam, you can go to my channel Omni Bio Entrance. In the playlist, see for next exam and you will find them there. So before coming to the next question, it is all regarding your enzyme kinetics and various types of inhibitions. I will tell you in brief what is each inhibition and what is the normal Michaelis-Menten curve. So this is the normal Michaelis-Menten curve for an enzyme catalyzed reaction. It is plotted as substrate concentration versus reaction velocity. And it is a hyperbolic curve. This is it. This is the hyperbolic curve, and the reaction rate, it increases rapidly at low substrate concentration, but at high substrate concentration it plateaus off. Okay, because the enzyme has become saturated with the substrate here. So that is the velocity maximum. That is this stage is the velocity maximum. And this is the Km of the reaction, or the Michaelis-Menten constant. That is the substrate concentration at which the enzyme reaction rate, reaction velocity enzyme, is half of the Vmax. Vmax by 2, that is your K. This is the normal Michaelis-Menten graph. Now the next is regarding the various inhibition of the enzyme. So first, this is the normal enzyme reaction. This is the enzyme. This is the substrate that binds to the active site of the enzyme, and it gets dissociated into products upon binding because of enzyme catalysis. Now there is an inhibitor called as the competitive inhibitor. It also has affinity towards the active site of the enzyme. So what this inhibitor does is, it will compete with the substrate for the active site of the enzyme. So the inhibitor, it will go and bind to the substrate binding site, that is the active site of the enzyme, and it will prevent the substrate from binding there. Okay. Now what happens to Km and Vmax upon this inhibitor binding? So in competitive inhibition, the Vmax is not changed, because if you increase the concentration of the substrate, because Vmax is attained at high substrate concentrations. So if you increase the substrate concentration, the inhibitor can be prevented from binding to the active site of the enzyme, because more number of substrate molecules will be there than that of the inhibitor. So the inhibitor can be outcompeted by the substrate and it can attain Vmax. So Vmax is not going to change upon competitive inhibition, but the Km increases. The Km value will increase. That is Km, I told you, it is the reaction rate at the substrate concentration at which the reaction velocity is half of Vmax. Now another thing I forgot to tell you about Km is that Km also indicates the affinity of the enzyme towards the substrate. Affinity of the enzyme towards the substrate. So if low value of Km means it has high affinity for the substrate. So low value Km, high affinity. Higher value Km, low affinity. Okay, remember that. It comes for next other inhibition reactions. Okay, now in competitive inhibition, the Km value increases because you need more substrates to reach the maximum velocity because there is competition between the substrate and the inhibitor. So in case of competitive inhibition, Vmax is not going to change, but Km increases or Km changes. Now coming to the non-competitive inhibition. So here the inhibitor binds to a site other than the active site of the enzyme. That site is called as the allosteric site. So this is the enzyme, this is the substrate molecule, this is the inhibitor. It will bind here. This is the allosteric site, this is the active site where the substrate is binding. So this is the enzyme inhibitor complex. So in this case, in case of non-competitive inhibitor, the inhibitor can bind to the free enzyme, that is the enzyme which is not bound by the substrate, or it can bind to the enzyme which is bound by the substrate also, because it is binding to a site other than the active site. Okay, so it can bind to enzyme substrate complex also. So now because of this non-competitive inhibitor, the Vmax is decreased because the enzyme is inactivated. Upon binding of the enzyme inhibitor to the enzyme substrate complex or the free enzyme, the Vmax is decreased because the enzyme is inactivated there. But Km is unchanged because the active site is not competed by the inhibitor, as in the case of competitive inhibition. And here in this condition, substrate concentration increase is not going to decrease the value of the Vmax. So is not going to overcome the decrease of the Vmax. That is what is happening in non-competitive inhibition. Now coming to uncompetitive inhibitor, see this diagram. So here the inhibitor binds only to the enzyme substrate complex. Only if the enzyme is bound with the substrate, the inhibitor will bind. It will not bind to the free enzyme. So here enzyme, it is bound by substrate, inhibitor binds to the enzyme substrate complex, that is uncompetitive inhibition. So here both Vmax and Km are reduced. So it results in a reduction of Vmax because the enzyme substrate complex bound to the inhibitor, it cannot release the product because inhibitor is bound there. So Vmax is reduced. And Km is decreased because the inhibitor bind to the enzyme substrate complex, which increases the enzyme's affinity for the substrate. Because it is having low Km. Low Km means higher affinity for the substrate. Okay. So changed Km and Vmax in the case of uncompetitive inhibition. Now there is a fourth type. I am not going to confuse you with that. It is mixed inhibition. It is a mixture of competitive and non-competitive inhibition. That much you understand. If I tell more, you will get confused. So now only you learn these three types of inhibition. Now coming to the question. Depicted in the figure are three possibilities of substrate, that is white triangle, conversion to the product, that is grey triangle, by an enzyme which is blue in color in the presence of different kind of reversible inhibitors. The corresponding interaction with specific inhibitors, black shades, are depicted schematically where the dotted double-headed arrow indicates a reversible reaction and a blunt arrow indicates inhibition. The Michaelis-Menten graphs right side of the figure shows plot in the absence of grey line and presence of inhibitor is the black line. Okay. So black line is the presence of inhibitor. Changes in Km and Vmax in each case are indicated. Based on your evaluation of the three modes of inhibition shown in the figure, correctly match the combinations. So this is your inhibitor. This is the substrate. The substrate is getting converted to the product here. This is the enzyme. So black line here, it represents an inhibitor, and gray line here, it represents the absence of inhibitor. That is your normal Michaelis-Menten graph. Now see the different types of inhibition based on what I have explained. So here if you see, this is the inhibitor plus enzyme. The inhibitor is binding to the active site of the enzyme and preventing the substrate from binding there. So the product is not formed. So this is a case of competitive inhibition. Because here the inhibitor, it will bind to the active site. So I told you in case of competitive inhibition, the Vmax is unchanged but Km changes. Okay. Km increases, or I can say Km increases. Now the condition where Vmax is unchanged. Here Vmax changes, okay. R. Vmax is unchanged. See here, the presence of inhibitor graph and the absence of inhibitor graph, Vmax is unchanged. But Km has increased in the presence of inhibitor. If it is somewhere here, then it is somewhere here in the presence of inhibitor. So 1 is R. 1 R you can see in A and D. Okay, now see the second one. Second one, this is the inhibitor, this is the enzyme, this is the substrate, this is the product. The inhibitor can bind to the substrate bound enzyme at a site different from the active site or the substrate binding site. That is this site. This is called as the allosteric site. Here to the free enzyme also it can bind. That is, it is bound here, and this site, that is the active site, is let free. It will block the formation of the product. So here that is a case of non-competitive inhibition. So in non-competitive inhibition, the Vmax is decreased or Vmax changes, but Km is unchanged. Okay, so that you can see in Km unchanged, Vmax changes, you can see here. Vmax is decreased here as I told you, but the Km is unchanged. It is the same value here for both the cases. So P is non-competitive inhibition graph and 2 is non-competitive inhibitor. Now see the third case. Third case, again this is the inhibitor, this is the enzyme, this is the substrate, this is the product. Enzyme substrate complex form enzyme and the inhibitor is bound. That is a typical case of non-competitive inhibition. Non-competitive inhibitor does not bind to the free enzyme. It binds only to the enzyme substrate complex and there is no production of the product. So it has a reduced Vmax and a reduced Km. So both Km and Vmax have changed. So if you see Q, 3 is Q. It is a case of uncompetitive inhibitor. So the answer here is D, 1R, 2P and 3Q. Then next one. In the plasmid vector map, tetracycline R, that is this one, it denotes tetracycline resistance and this denotes ampicillin resistance gene. SAL1 restriction enzyme digested DNA fragment containing the gene of interest is ligated to the SAL1 digested plasmid vector. So you have a SAL1 digested gene of interest or a DNA fragment and you digest this plasmid also with the SAL1 and you ligate it with this fragment. So you will get the ampicillin resistance gene being damaged, tetracycline resistance gene intact, and your gene of interest ligated to the ampicillin resistance gene. Okay. This was then transformed into strain of E. coli that is sensitive to both the antibiotics. So they have tested the E. coli and have found it is sensitive to both the antibiotics. Okay. The desired transformants, that is containing the recombinant plasmid, were obtained by plating this reaction on tetracycline containing nutrient agar. So once you this is the transformant, and you will find that the transformant is sensitive to ampicillin in the medium, but it is resistant to tetracycline because tetracycline gene is intact and the ampicillin resistant gene is damaged. So in the presence of tetracycline containing nutrient agar, the transformants will grow. But our question is not that. They have asked an appropriate control for the above experiment, a control, okay, that can demonstrate complete digestion of the plasmid vector by SAL1. So in certain cases, when you do digestion of the plasmid or any DNA fragment, there are chances that you get an incomplete digested vector. So what will happen to this incomplete digested vector is that there are chances of self-ligation. Okay, so the vector will ligate to itself. It will not take the gene of interest and it will not form the recombinant. It will ligate on its own. So your ampicillin resistant gene will not be damaged. And such incompletely digested vectors, they can grow in ampicillin containing medium. Okay. Now come to the options. Ligation of BAM-H1 digested plasmid vector with SAL1 digested insert and transformed into E. coli followed by plating on ampicillin containing nutrient agar plate. So if you, ligation of BAM-H1 digested plasmid vector, that is tetracycline resistance, you are digesting, and then you ligate it with SAL1 digested gene of interest. Okay, that you cannot ligate because one is digested with BAM-H1, other is digested with SAL1. You cannot ligate both of them because they will not have compatible ends to join. So option 1, you are not going to get a correct ligation. So that's not correct. Here you have to see whether the vector is completely digested. Ligation of SAL1 digested plasmid vector alone. So you take the plasmid vector, digest it with SAL1, then you ligate the plasmid vector. What do you get? You will get the same vector. You are not putting any gene of interest. Okay. And transformed into E. coli followed by plating on ampicillin containing nutrient agar plate means naturally they will grow in the ampicillin containing medium, because after SAL1 digestion you are ligating the vector, you will get ampicillin resistance gene intact. It is not going to be damaged. So it will grow in ampicillin containing nutrient agar plates, and you will not know whether there was any complete digestion of the plasmid. So B is also not correct. Then unligated SAL1 digested plasmid vector. Okay. So you take this plasmid, digest with SAL1, and you don't ligate it. That means you get a linear fragment of DNA or linear fragment of the plasmid and transformed into E. coli followed by plating on ampicillin containing nutrient agar plate. Okay, now that would be a very good choice, because now when you digest the plasmid with SAL1, if it is not fully digested, I told you there are chances that it will ligate back and you will get colonies on ampicillin containing medium. Now if it is completely digested with SAL1, it is completely digested with SAL1, it will not ligate on its own. You have to put the ligation mixture there, only then it will ligate. Okay. And you transform E. coli, it is not going to transform E. coli because it is a linear plasmid. And colonies will not grow on ampicillin containing nutrient agar plate. So you can conclude based on whether colonies are appearing or not, whether it is fully digested or partially digested. Okay. So C is the answer. Unligated SAL1 digested plasmid vector alone transformed into E. coli followed by plating on tetracycline. Tetracycline naturally they will grow because it is not damaged. It is not digested with SAL1. It will not tell you whether it is because the SAL1 digestion is complete or incomplete. So D is also not the answer. Now next one. A small group of mammals that consisted of closely related kins migrate into a new ecosystem. Despite inbreeding, the population is stable for many generations. However, due to sudden appearance of man-made geographical barrier, the group split into two groups P and Q. So you had a large group of mammals which are closely related kins, and because of the man-made geographical barrier, they separated into two groups, that is P and Q. Now what happened as a result of this separation? The population soon begins to decline, and conservationists, they decide to take action to increase the population. So what strategy they have to apply for increasing the decline in the population? So first one, to have a separate breeding programs within group P and group Q. So separate breeding programs maintain isolation and reduce genetic diversity. So that will not be a very good option. To create barriers to avoid predators. So here they have not told anything regarding the predators in the ecosystem. So that's not the correct answer. To move all individuals in one group to the location of the other group. If you do that, there will be overcrowding, competition, etc. So that also is not the right answer. To create corridors to facilitate migration. Yes. So as a because if you create some small kind of a hole or something like that, just for you to understand, I am telling, the P and Q can migrate through this hole and undergo mating, and there will be increased genetic diversity and population stability and growth. Okay. Or you can create a bridge. If there is a sea, a water body there, you can create a bridge so that the P and Q can migrate through this bridge and undergo mating, and there will not be decline in the population. That is what is meant by to create corridors to facilitate migration. So D is the answer. Then a particular microbial pathogen that infects human has cell wall composed of peptidoglycan layer. An infection from this pathogen can be easily treated with antibiotic that inhibits cell wall synthesis. A hand wash containing this antibiotic was popularly used. In subsequent years, resistance to the antibiotic emerged in this pathogen. While one group evolved a modified cell wall, the other group evolved to survive without a cell wall. This type of selection is. So there are three types of natural selection: disruptive, stabilizing, directional. Disruptive means when the intermediate, both the extreme traits are favored over the intermediate. Sorry. Stabilizing means where the intermediate phenotype is favored, and directional means only one extreme phenotype is favored over the other. So in this case, both the extremes are there. One extreme is evolved with modified cell wall and the other is without the cell wall. The intermediate is having the cell wall composed of peptidoglycan. So this two different extremes are favored in this type of selection, so it is disruptive. Answer is option A. Now, the table shows the presence of photosynthetic pigments in bacterial and algal group. So, this is photosynthetic bacteria, this is the algal group. All eukaryotic chloroplasts are derived from a common ancestor. It is well known that it was the result of a single endosymbiotic event. Assuming loss of gene or genes during the course of evolution, the correct phylogenetic tree is. So this is the data that we have. This is photosynthetic bacteria and this is photosynthetic algae. So photosynthetic bacteria gave rise to Prochlorophyta and Cyanophyta. Prochlorophyta had chlorophyll A and B, and Cyanophyta had A and phycobilins. Whereas photosynthetic algae, gave rise to Glucophyta, Rhodophyta, Chlorophyta. Glucophyta, Rhodophyta, Chlorophyta, they all had chlorophyll A. But phycobilins are present only in Glucophyta and Rhodophyta, whereas Chlorophyta doesn't have phycobilins, it has chlorophyll B. Now if you see this option C, you can see that both bacteria and algae are put together, which is not correct. Here in D, they have shown that Prochlorophyta gave rise to Chlorophyta. That is not correct. How can an algae rise from bacteria? So that is also not correct. Now B, if you see, again the same mistake repeated. Prochlorophyta, Chlorophyta arose from bacteria. No. So what is left out? A. So A, this is bacteria and this is algae. So from bacteria gave rise to Prochlorophyta and Cyanophyta. See here. A, the Prochlorophyta is having A and B chlorophyll. Cyanophyta is having A chlorophyll and phycobilin. So they both are rising from the same ancestor, and chlorophyll A and phycobilins in Cyanophyta. But in photosynthetic algae, Glaucophyta and Rhodophyta, they are from the same lineage, whereas Chlorophyta is from a different lineage. So A is the answer. Chloroplasts of certain algal taxa such as Cryptophyta are surrounded by 4 membrane envelope. The most likely reason for the origin of 4 membrane enveloped chloroplast structure. We discussed that in session 1 question paper, and they have told it is because of secondary symbiotic event. So secondary symbiosis where a photosynthetic eukaryote was engulfed by another eukaryote. So that is the answer. Answer is option A. Then camels have 3 different subclasses of Ig, IgG1, G2, G3, of which G2, G3 are structurally different from the human IgG. G2 and G3 are different, but G1 is similar. A researcher purified camel IgG1 and G3 and carried out gel electrophoresis that separates polypeptides based on their molecular size. An IgG antibody purified from human serum is electrophoresed on the same gel. A schematic of the observed band pattern on the gel is shown in the figure, wherein lane M depicts the molecular weight marker positions in kilodaltons. R means the samples are treated with reducing agent, that is beta-mercaptoethanol. And this beta-mercaptoethanol, it has a property that it will dissolve the disulfide bonds that hold the polypeptide chains. NR is non-reducing was used. That means your disulfide bonds are intact in NR. Based on this information, the correct structure of camel IgG3. So this is the gel. This is the molecular weight marker. This is NR means without any non-reducing agent, and R means because of the presence of reducing agent. So human IgG, camel IgG1. They have already mentioned in the question, they are almost similar, but camel IgG3 is different from human IgG. Now this is your antibody structure. This is the heavy chain. These are the light chains. These are the disulfide bonds that I have drawn here. Okay, in the presence of reducing agent like beta-mercaptoethanol, the disulfide bonds will be broken. So you will get 2 light chains like this and 2 heavy chains like this, which are not bonded by disulfide bonds. These 2 light chains, they will have the same molecular weight and they will be present as a single band. These 2 heavy chains, they also have the same molecular weight and they will be present as a single band. Okay, now see this. This is NR, this is non-reducing, so it is the intact antibody, that is this. That is the size of that. Here also NR is there. Here NR is slightly lower molecular weight than IgG and IgG1 in Ig3. Okay. Now this is your 50 kDa that corresponds to your heavy chain, and between the 20 and 30 it corresponds to your light chain. Similarly, camel IgG1, this corresponds to 2 heavy chains and this corresponds to 2 light chains. I thought 2 heavy chains are there, 2 light chains are there. So that is what this corresponds to. So here only the heavy chain, 2 heavy chain bands are present, but light chain bands are not present in Ig3. So it is 2 heavy chains bonded by disulfide bonds. So the answer here is option A. Then interspecies interaction are well known among soil dwelling bacteria belonging to the genera Bacillus and Pseudomonas. When co-cultured, Pseudomonas chlororhabis competes for space with Bacillus subtilis, infiltrates their colonies and kills. So that is, killing is the key word here. Killing of vegetative cells of Bacillus subtilis by Pseudomonas chlororhabis requires physical contact and the type 6 secretion system to inject the toxins. Okay, that is the mechanism how it will kill. That is all what is mentioned by that particular sentence. Now Bacillus subtilis, what it does to prevent the killing is it will undergo sporulation and escape to avoid killing by Pseudomonas chlororhabis. On the other hand, when Pseudomonas chlororhabis are co-cultured with Bacillus amyloliquefaciens, colonies have distinct zones with no physical contact. In this scenario, chlororhabis utilizes a different secretion system, that is T2SS, to secrete secondary metabolites that inhibits the growth of amyloliquefaciens. Okay, so in the first case with Bacillus subtilis, it is killing the Bacillus subtilis, and in the second case it is inhibiting the growth of amyloliquefaciens. Bacilline, a bacteriostatic compound, that is bacteriostatic means it will inhibit the growth, is produced by Bacillus amyloliquefacien that antagonizes Pseudomonas chlororhabis. So in the first case, Bacillus subtilis used sporulation to avoid killing, but here Bacillus amyloliquefaciens is not killed. Only inhibition of the growth is there. So to counteract it, Bacillus amyloliquefaciens is also producing a growth inhibitor, that is bacilline. They both are not killing themselves. In second case, that is Pseudomonas chlororhabis and Bacillus amyloliquefaciens. So what kind of interaction is there? So in the first case, it is predation, because chlororhabis and Bacillus subtilis, it is predation because chlororhabis kills Bacillus subtilis. Okay, so it is predation. So A is the right answer. The second is competition, because the interaction between chlororhabis and amyloliquefaciens, they compete for the resources. Okay, that is why it is not producing any toxins to kill Bacillus amyloliquefaciens. They are only producing growth inhibitors. So it is a case of competition. Now commensalism means one organism is benefited whereas the other is not affected, neither positive way or negative way. Then mutualism means both organism benefits, which is not the case here. Neither is commensalism. So predation and competition is the answer. Mitochondria are present in species P but absent in Q, R and S. So mitochondria are present in species P means P must be eukaryotic. Q, R, S it is absent means those three are prokaryotic. Now see for prokaryotes and eukaryotes here. So first one, slime mold, it is eukaryotic. So mitochondria is present in P. Blue-green algae eukaryotic, prokaryotic. Eubacteria again prokaryotic, mycoplasma prokaryotic. So they represent Q, R and S. So first one is right. Whereas if you see B, it is blue-green algae first. That is P is representing blue-green algae. It is prokaryotic and it will not have mitochondria. So B is not correct. Third one, it is plant. Okay, it is having mitochondria. Slime mold again mitochondria, but they have mentioned QRS as not having mitochondria. So C is also not correct. Mycoplasma again, it is a prokaryote. P is not a prokaryote. So D is also not correct. A researcher has cloned a bacteriophage gene P into a bacterial vector under the control of phage promoter. To express this gene in bacteria, a particular strain of bacteria that carries phage RNA polymerase is used. The phage RNA polymerase expression is turned on, in turn can be induced by addition of an inducer to the medium. Rifampicin, it specifically inhibits bacterial RNA polymerase and not phage RNA polymerase. Chloramphenicol bind to bacterial ribosomes and block protein synthesis. The condition where the researcher will be able to express maximum amount of protein P in bacteria. So here what the researcher has done is he has cloned a gene P that is from bacteriophage into a bacterial vector, and the gene P is under the control of the phage promoter. Okay, now the promoter is phage. From the phage, the gene is from the phage. So if you have to add phage RNA polymerase so that it will bind to the phage promoter and it will result in the synthesis of protein P. So to gain that, what he has done is he has used a special strain of bacteria that carries phage RNA polymerase. Okay, now this particular bacteria having phage RNA polymerase is also having bacterial RNA polymerase. Now what the experimental will do is he will add rifampicin there, so that bacterial RNA polymerase is not active or the bacterial RNA polymerase is inhibited. So only phage RNA polymerase will be there. It will bind to the phage promoter. Gene P will be transcribed. Okay. So protein from gene P will be maximum produced. Now for the phage RNA polymerase to be active, he has to add an inducer into the medium. So he has to block bacterial RNA polymerase by adding rifampicin, as well as add an inducer so that the phage RNA polymerase will be activated. Okay, now coming to the chloramphenicol. What does chloramphenicol do? It will bind bacterial ribosome and block protein synthesis. Now if he add chloramphenicol into the medium, what this chloramphenicol will do is, RNA polymerase is only for transcription, only mRNA will be produced. Now mRNA has to synthesize proteins. For that bacterial ribosomes are needed. Now if you add chloramphenicol, it will bind to bacterial ribosome and block protein synthesis. So naturally protein P will not be synthesized. Now see the options. Addition of inducer and rifampicin. Yes, I already told. Inducer addition, it will activate phage RNA polymerase. Rifampicin addition, it will inhibit bacterial RNA polymerase. So phage RNA polymerase will go there, bind to phage promoter. Gene transcription will take place. Then bacterial ribosomes will convert this mRNA into proteins. So A is correct. Addition of inducer and chloramphenicol. You are adding chloramphenicol, bacterial ribosomes will not be active. They will block protein synthesis. You will not get expression of protein P. So B is not correct. Addition of rifampicin and chloramphenicol. What is the fun? Rifampicin will inhibit bacterial RNA polymerase. Then no protein synthesis because chloramphenicol is there. So that is also not correct. Addition of only rifampicin. It will block a bacterial RNA polymerase, but there is no inducer to activate phage RNA polymerase. So naturally you will not get maximum expression of P. So that's it. Thank you for watching my channel. Please do like, share and subscribe.